1953 was a busy year for scientists. While Stanley and Urey were trying to cook up amino acids in a test tube, Watson and Crick had been working assiduously on the structure of DNA, something which had eluded scientists for decades. On February 28, 1953, Francis Crick strolled into the Eagle pub in Cambridge and made a profound declaration. He said, "I'd like a beer." Just kidding. He really said, "We have found the secret of life." Watson and Crick's declaration was no understatement. They'd made the amazing discovery that the DNA molecule was comprised of two strands of nucleotides bound together in a helix, going in opposite directions, like a spiral staircase. It's time for a brief biology lesson, so that you can make sense of the magnitude of this discovery. You see, we have 46 DNA molecules, which join to form 23 pairs of chromosomes in the nucleus of every cell in our bodies. Relying on Watson and Crick's breakthrough, it soon became evident to scientists that the amino acids and proteins necessary for life are made from the coding found on small, discrete sections of our chromosomes, called genes. These genes contain a set of step-by-step instructions, perfectly sequenced to build protein and amino acids, each at the exact time and in the correct sequence. Back in the early 20th century, evolutionists, like Oparin and Haldane, mistakenly believed that just as inorganic chemical compounds are made by atoms randomly bumping into each other under the right set of circumstances, so too, life-forming amino acids, and hence proteins, could be made by the same blind, unguided process. However, the coding found on our genes shows that protein synthesis is definitely not an unguided process, but one which has a reliable and repeatable set of instructions or code to guide the process. Recently, scientists have been able to create a few simple proteins by stringing together amino acids in the laboratory for medical and industrial use. However, firstly, the technology largely depends on harnessing the existing coding of DNA found in living organisms, rather than the de novo creation of a new technology. Secondly, and most importantly, and something I'd like you to think about, if scientists with advantage of 21st century technologies can make a few small chain proteins, then it would be hard to conceive how the blind forces of nature, without the assistance of science, could possibly make sophisticated proteins billions of years ago, in the primordial era.